A new podcast from the University of Michigan's College of LSA. This is How to Science with Professor Monica Deuce.
Discovering that scientists are humans and not robots. Here's a protocol for listening to this podcast. Step 1, turn down the Bunsen burner. Step 2, turn up the volume on the speaker by your beaker. Step 3, start listening. The first time I met Trisha Wittkopp at the University of Michigan was at the Teaching Academy, a program run by the College of LSA that helps young professors like me be better prepared for teaching. So at that point, I knew two things: that Trisha had taught genetics, the class that I was supposed to teach, and that the students really loved her. Genetics was taught in this huge classroom, like a Roman amphitheater. And maybe it's because I'm Italian and I studied Roman history, but that classroom looks like the place where gladiators would fight lions and other beasts as the crowd would cheer and sneer. I was really intimidated at the thought of teaching genetics, and I was really reassured and inspired when I heard Trisha talk about how she tackled genetics as a young professor. So in my mind, she was this legendary figure who had fought with love, conquered the hearts of the crowd, and helped them discover new things. I pretty quickly fell in love with her, and I can't imagine anybody not falling in love with her and her lab after they hear about their crazy lab tradition of donut data days, lab Olympics, and also Trisha's overall awesomeness. Hi, Trisha.
Hi, Monica. As you know, I'm a professor here in the departments of ecology and evolutionary biology and also molecular cell and developmental biology.
So I want to ask you a question that became evident as I was looking through your website. You have a lot of pictures with your lab. You have lab Olympics. There is a picture where you're holding a giant turkey and where you had your lab over for Thanksgiving dinners. And it's really clear from those pictures that you guys have a lot of fun in the lab, and you seem to take the idea of science done as a team as a core principle in the lab. So can you tell us a little bit more about that?
Sure, absolutely. As much as anything can be by design as a group of people come together over the years, the desire to make science fun and the lab atmosphere fun was actually really fundamental from the beginning. Before starting my position, I read a lot of advice books about how to set up the lab, and they talked about having a lab motto. And the one I came up with was science is fun. Science should be fun. I mean, that's what brought me into science. It's what I think brings us all together, is this passion for knowledge. And the more we can have fun along the way, the better. I think that doing things like the Lab Olympics, where we get a chance to compete on these rather esoteric skills that we've honed day to day in the lab, or we have an annual canoe trip, or we've gone and play volleyball, and we have various lab gatherings, allows the people in my group to build the personal relationships that really support their science and make it easier for them to ask each other for advice or help or speak more freely about when something's not going right. I think it helps reduce the competitiveness in the lab. There's so much, I would say, competition in science and also critical assessment of your work that happens through the review of papers and grants. And there's lots of negativity sometimes that comes through that the hope is that the lab environment becomes sort of a safe space for exploring your scientific interests, but also where you can take a risk intellectually and be supported by those around you.
In many ways, it's not a common thing in science. The idea that a lab would be fun was almost a taboo because it's almost like you don't take your science seriously, which we know now it's not true. So how does each member of the lab, especially we have people from all different cultures and countries, how does that play in making this atmosphere fun and unique?
Well, I would say certainly everyone's individual strengths are welcomed and supported. Most of our lab gatherings are a potluck of some sort, and so everyone's bringing various dishes. And so one of my first graduate students was from Turkey, And so I remember she made this lentil soup that was amazing. It's a great metaphor, I think, for the way the lab works, is each one bringing their own expertise in something they love and everyone else sort of being open to and celebrating that. In fact, we have a lab cookbook now, or sort of recipe book that we've assembled to share all these recipes, because it's a great way to experience new things, and then we find new things that we love ourselves. So now when I have this habit to make lunches easier when life is busy, I make a big pot of soup and I freeze all these individual packages. One is the lentil soup I mentioned. So every time I make it, I think of Gizem, my first grad student. In addition to good food for me, it's sort of the memory and the reflection on people that make the lab what it is. I do want to be careful to say that I think that where this idea and philosophy came from was actually my own graduate lab. So if I give a shout out to Sean Carroll, it's interesting that you said that sometimes you might have that view that if you're having too much fun, you're not taking your science seriously. Sean, my grad advisor, was the most down-to-earth person. He quoted Animal House at lab meetings. He always had his feet up on the desk to think. He was a Howard Hughes investigator. He's now the vice president for education and outreach at Howard Hughes. He's undeniably very successful and takes his science very seriously, but yet the atmosphere in the lab was always one of fun and support. One thing that we did there that I do in my own lab is we celebrate various successes scientifically as well. So one of the smallest scale is our donut result. When somebody has a significant step forward on a project, something that might make a figure in a paper or something, they bring in donuts and we all sort of enjoy the donuts and celebrate. Then the larger milestones like a grant or a fellowship or someone's first author paper, those get recognized with champagne. And then we have a collection of champagne bottles as a visual reflection of the lab's accomplishments. So I think that having that experience living in a lab, that was fun myself. And wanting that for my own group helps. And then I have to be very, very quick to say that ultimately it's the people in my lab that make that a reality. That's not something you can dictate to say lab must be fun. I think the best I can do is create an environment that allows it and supports it.
Was the lab culture in the Carroll lab something that attracted you to the lab? So this might be a good time for me to ask you how you got to graduate school.
Sure. Maybe I'll back up quite far, which is to say that I was not a kid that grew up chasing bugs or thinking about science, right? It was like a really good high school teacher that got me hooked on genetics. I was raised by a single mother that didn't have a college education.
Oh, wow.
So I was just looking forward to going to college and trying to find a way financially to do that. I didn't know that grad school existed. The idea that you could get a PhD, which again, I didn't know what that was, or be a scientist as a career was not at all on my radar. That was so not in my family's worldview. So when I got to the University of Michigan and knew that I wanted to study genetics, I had to figure out what that meant. I gradually learned that there may be a way to do this as a career, and then was told, well, you need some research experience. So like many undergraduates, I went through all of the web pages of the faculty in the biology department at the time and looked for anyone that had the word genetics on their page and sent them an email. I got a response from one person. This was a new faculty member who I jokingly say probably didn't know he didn't have to respond to all of those messages. But fortunately for me, he did. And he, invited me to come in and learn about what he was doing. This was a man named Greg Gibson, who's at Georgia Tech now. Still one of my cherished mentors, I would say, to this day. And what he was studying, as he described it, was how cells become different, right? So all the cells in the body have the same DNA. And it's really fascinating to think about how do you create different structures? Why is an eye cell and a heart cell, and a blood cell, when they all have the same DNA? And so as I learned about gene regulation, I thought that was really cool. And then one moment in that lab crystallized for me that I wanted to go to grad school. I was doing the experiment that Greg told me to do, to be blunt. Obviously, with undergraduate students who don't have a lot of research experience, there's a lot more guidance in those early projects. And so he had asked me to cross flies together with different types of eyes and decide how rough the eye looked, right, on a scale of 1 to 10. And so I was doing that and keeping all my careful notes. And I was noticing that the eye colors weren't exactly what I expected them to be. At the same time, in parallel, and maybe this is a good example of research and coursework merging, I was learning about things like dosage compensation in my classes. And I came up with this idea that if the gene I was studying, instead of being on an autosome, was on the X chromosome, that might explain all the weird eye colors. And I went to Greg and I said, I think maybe this could be going on. I've been keeping track of eye colors too, and this is what I see. And he said, Trisha, I think that that's an interesting hypothesis. I think that the people that determined where that gene was are really experts in the field, and I don't think they would make the mistake of putting it on the wrong chromosome. But
there's only 4 and one
but
is not
that's
really
right. And the X has such different inheritance patterns, right? So he said, but if you want to test your idea, this is how you do it. And he showed me how to do a fluorescence in situ hybridization, and I did it, and I was right. And in the grand scheme of science, it's not very important. But for me in my career, that ability to observe something that I felt on my own, like I just noticed it, come up with a hypothesis, test it, and then to have that validation that I could do that was just, I mean, addicting is the word that comes to mind, right? From that, there was nothing else I wanted to do with my future at that point. And so that was the moment that I knew I wanted to go to graduate school. Fortunately, having Greg as a mentor, somebody who was in the lab with me, sitting next to me at the microscope day in and day out, and we would talk about science. And all of that, those conversations actually led me to apply to the University of Wisconsin and to work with Sean Carroll. And Greg, I know, had crossed paths with Sean at a meeting I had found out later and had mentioned me to him. So Sean was sort of looking out for me. And within a few days, going back to the genesis of this question, you asked about sort of the atmosphere. Yeah, it just felt like home. That's all I can say. Within a week or so, I knew that that was the right place for me. And it was an environment that was filled with about 20 people that were all very smart, very kind, very funny, who all had different training that were brought together to work in this interdisciplinary science, right? And so it was this environment where, like I said, there was a sense of community, culture, openness, and sharing with a mix of expertises. I'm not sure that's a word, but that, yeah, created a combination of freedom and support that was really great for me.
And it seems like it's almost the ideal science experience because, you know, I can see how going through the process as an undergrad teaches the way, in first person, the way science does work, that it's okay to challenge things that you thought were decays and to ask new questions and then to rewrite maybe, you know, a new dogma that then gets challenged again.
Yeah. I mean, looking back on it, I think that the way Greg handled it in saying, I don't think so, but here's how you figure it out. And giving me, you know, the freedom and the tools to test that. I mean, I'm not sure everyone would have said that. I think that was really just a pivotal moment for me that put me on a track to where I am. There's still moments, right, that, you know, now I've been a professor here at Michigan for 12 years, and I've had an opportunity to visit many different places in the world, different institutions, you know, invitations for talk at meetings and seminars. And there are moments that I look around and think, you know, how did I get here? Right. So I visited Harvard, actually, and they put me up in the Harvard faculty club as they do seminar speakers. And I looked through the guest book, you know, and it was like Colin Powell. And it was just one of these moments of this girl who grew up in a very blue collar city raised by a single mother. Like, how am I here? And it feels normal, right? Because it's step by step that day to day, I don't have those reactions, but every once in a while when you step back, it's, I don't know, I have a hard time putting it into words.
No, it makes sense because the day-to-day sometimes can be very overwhelming, and then one day you find yourself with a major accomplishment, and then sometimes it's just easy to focus on all the little things that went wrong than to just look back and say, oh, wait, I am here today. So, and that's why it's so important to then celebrate the accomplishment with like the donut.
Yeah. I think the other nice thing about the donut result is it's intentionally, each person decides what's a donut result for them.
Oh, really?
Right? I don't decide. It's not my judgment.
Cool. Okay.
It's not anybody else in the lab deciding when you've accomplished enough to earn donuts. Sometimes it's something that objectively maybe doesn't advance the project that much, but has been technically so
like
challenging.
That cloning that
right?
didn't work.
That cloning, that's right. The cloning you beat your head against the wall for 9 months and it finally works. Or our first transgenic flies or, you know, things like that I think it's just a way to celebrate, like I said, the short-term successes in a way that is independent of anyone else's assessment. You know what I mean? There's not a reviewer saying, no, you can't have donuts for that. It's your own sense of accomplishment.
So What was it like to be in the Care Lab? What did you do there, and how did that make you grow even more as a scientist?
I think some of his unique qualities that have made him as successful as he is, and that I hope I have learned some of, are sort of a clarity of thought and communication, the ability to synthesize information, the ability to see through the forest of possible sort of experiments to find the one tree or the one approach that is actually going to answer the question, and then to put all your energies into that one thing. So I think that combined with being down to earth and not really having much of a hierarchy in the lab, everyone's individual opinions and viewpoints were valued. Whether you were an undergraduate researcher or the senior postdoc in the group, there was respect for everyone's thoughts and ideas. And to some degree, the freedom that he allowed, that there wasn't a lot of hand-holding. And to be blunt, that wasn't the best fit for everyone. There were a number of grad students that came and ultimately decided to leave the lab. For me personally, there was that freedom and independence, though, that I was allowed to do something that maybe he didn't know I was doing. Sometimes it worked great, and other times maybe it didn't. And that was okay, that the lab and the atmosphere could support that.
Your project was on fruit flies.
Yes. Actually, going back to that first question that Greg asked me, how do different cell types become different? Over the course of my career, that's expanded to how do individuals of the same species become different, and how do different species become different? So all those levels. And again, that geneticist core in me says, what's the genetic basis of that? The answer to the cell type question comes down to gene regulation. So each gene has, I describe it as like a dimmer switch that would control your lights, right? It can be on, off, or on to different degrees, and that's true for every individual cell. So the combination of cells that turn on a gene and how much of that gene they produce are what gives the cell largely its properties. And so changes in those switches underlie differences between individuals of the same species and between species. So as a graduate student, I chose pigmentation in Drosophila as a case study to study that. So pigmentation is one of the most variable traits in actually most plants and animal species. And one of the advantages that allows is, well, number one, it's not usually essential for life. So you can manipulate it without hurting the organism, right? You can still raise them in the lab. And there's a lot of natural variation for it. So you can study the genetic mechanisms underlying it. What I learned was that actually it's often different due to changes in gene regulation. So I sort of phrased it here as a going in looking at gene regulation, but really that was what came out of the genetic analysis.
So with all this positive experiences in academia, was your aspiration to become a professor, or did you consider, different kind of naturalist careers?
Yeah, definitely not the naturalist piece. I joke with my husband, who ended up being a high school biology teacher for a while, though now he's an academic advisor here. He was also a biology undergraduate major here at Michigan with me, and he had taken all of the ology courses, if you will. And he, if we go on a walk, he's the one that could identify the tree and what type of frog that was that croaked. And I sort of joke that I'm just lucky if I could say that that's a grass, or that's a bush, or that's a bird, that's got wings. Well, it's not always a bird, but you're right. So, but I can tell you about how that flower was patterned in its meristem as it grew.
True geneticist.
That's right. And I think the geneticist at the core is what drives me. Pretty much from the time I decided I wanted to go to graduate school, being a professor was really the only thing I was focused on. And I'm not sure that's, well, that is what it is, right? That was my reality. That was what I wanted to do.
So was your postdoc then still on pigmentation?
No, for my postdoc, I had taken the advice to go and do something different.
Okay.
Yeah. Which I remember in thinking about postdocs was really super stressful, right?
Yeah.
I felt like I was being told two things. The first was you should go learn something new as a postdoc. And the other reality was that what you do as a postdoc is what you're going to do in your lab for the rest of your life, for the next 20 years or something. And so it felt really hard for me to think about, how do I choose something that I know I'm going to like to do when I haven't done it? Like explicitly, I haven't done it, and that's why I have to consider it, right? With the hindsight I have now, I would say that was way too much pressure and not true. Like, that's not the way it works. But I decided to go and work with a guy named Andy Clark at Cornell. I started out on a project that was part of a larger collaboration he was involved in, looking at human genetic sequence data
I didn't know that.
to analyze
Okay.
stuff. There's nothing on my CV that would ever reflect this. But it gave me, I spent probably a good 6 to 8 months learning to program. At the time, it was a Perl programming book, learning to work at the command line and getting the skills to even handle the large data sets and move things around, Then one day, I was at a seminar and the talk was about, I don't even remember, something with mouse genetics. And something that he said about RNAi triggered a thought for a project idea I had in grad school. In grad school, I considered doing this experiment where you could cross 2 species together and use RNAi, which is RNA interference, to knock down one allele or the other. And my thinking was that you could sort of march across the genome and find genes that had changed between the 2 species in this way. I never did the experiment. I don't think it's doable. But what it made me think about was the allele specificity. And so what I realized in that moment during that seminar was that if you cross 2 strains together and make these hybrids, so I've mentioned the gene expression being controlled by switches. There's really 2 components to that. One, maybe we should take that analogy further, would be the switch on the wall, and the other would be the hand that needs to turn the switch. So switch on the wall would be the cis factors, and the hand would be the trans factors. So those switches get inherited with the gene. So in hybrids, you can look at the RNAs that come from one allele or the other, and if there's a sequence difference between them, you can figure out which actual molecule came from which copy of the gene.
Mm-hmm. So if you think of that as a sandwich, one piece of bread comes from one side, one piece of bread comes from the other. The piece of bread are the switches.
Yes. Yeah. Each piece of bread has its own switch. So if if those 2 switches are not working the same, you can get different amounts of RNA coming from each of the alleles. If you had a way to measure that, you could directly test for cis regulatory differences. When a gene changes expression, where in the genome are the changes that cause that change in expression? Are they in the switches, the cis regulatory sequences, or the things controlling those switches? And so as a grad student, answering that question for one gene took me 3 years of a lot of work. And in this second half of the seminar, I realized that if we had a way to just track which transcripts came from which allele, we could do this quickly and easily. And in my lab that I was in there, in Andy Clark's lab, he had a tool called a pyrosequencer that was used for genotyping DNA. And I realized that we could adapt this to looking at RNA levels and look at the relative expression of the 2 alleles.
So you were working on something completely different.
Yep.
Learning how to code.
Yep.
In front of a computer, away from your fruit flies.
Yes.
And then you went to a seminar that maybe you could have also never gone that day.
Yep.
Sometimes we have to. Sometimes I don't want to keep coding.
Yeah.
And then as you're listening, something that's not related to your research trigger back a thought that you had a few years back and
Changed the course of what I was doing.
Of your postdoc.
Yeah. And so within that second half of that talk, I remember the whole idea kind of forming.
That sounds like a donut moment, but not for data.
For ideas. Yeah. The person sitting next to me at the time was a fellow lab member, Brian Lazaro. And the talk ended. I was like, Brian, I think if we did this, we could do that. And he's like, yeah, I think that'll work. And so I remember running upstairs to the lab, and Andy was there, my postdoc advisor. And Andy's pretty quiet, and I was really charged, right? I was like, Andy, I think, and we could use the pyrosequencer, and we could measure this, and we could blah, 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 and then we could sort out cis and trans differences, and we could do this for lots of genes, and it would happen quick. And I remember going, yeah, I guess you can try that. And I would be very careful to say here that and my experience in Sean's lab, one thing that both of those labs had that allowed me freedom was they were well funded. They were large enough, they were established enough that they could let people in the lab take risks without really compromising the stability of the lab as a whole. And so with the green light, I did this. It worked out. It ended up being published in Nature. Now this approach is used by lots of people in different fields. And it really changed, I think, it definitely changed the focus of my postdoc. It became one of the first things I used in my own lab to get it up and running. And then I would say inspired in part by my experience in Sean's lab of taking a project idea wherever it leads. We've adopted a new model system with yeast. New to us, I should say. It's a very classic genetic model system. But what adopting that system has done is allow us to systematically study the effects of new mutations, which are changes in DNA that underlie all variation. and look at how these new mutations affect gene expression, which is, again, not only important for genetic variation and evolution, but also diseases like cancer, which are purely driven by new mutations, many of which affect gene expression. So I view that part of our current work as really basic biology, right? How do mutations affect gene expression? Although we often frame it in how does this create raw material for evolutionary change? So we'll compare the effects of these new mutations to the effects of variation in natural populations, and that allows us to tease out what contribution selection has had versus mutation, which is a fundamental question in evolutionary biology.
So one thing that it's cool to me as I listen to you tell your story is that you're really in love with genetics. Throughout your career, when you weren't really using it as much as you wanted, you found a way to do it. And then you were undergrad at Michigan to go back and now be the person that's teaching genetics instead of taking genetics, and what your love for that subject meant and how that drove you to change it.
Yeah, absolutely. So as you say correctly, genetics is really what drew me into science in general. It's what drew me through my PhD, and it's still at the core of everything I do in my research program. So I was really excited to teach genetics when I came here, which was not the reaction most people have. At the time, I think they really needed to find somebody to teach genetics. That may have factored into my hiring, I would suppose. But it was my first year. It was the first time I had taught. So I had not had much teaching experience. I will credit being married to a high school teacher. So we had a lot of biology teaching magazines laying around the house that the societies put out. And I could bounce ideas off of him, and I did, for ideas for the class. And at first it was a little surreal, honestly. As you say, I was an undergraduate. I took this exact class in the same room that now I'm standing up there teaching it probably about 7 or 8 years later. So it was very surreal. The person I was co-teaching the class with, Steve Clark, who was very generous in sharing all of his materials, which formed the beginning of all of my lectures. So the first time through a class, I think you take all the help and support you can get and take advantage of previous efforts. So that was also a little surreal, from being his student in his class to now teaching with him. The course pack of problems that we had at the time that we gave to the students was the same course pack of problems I had as a student. So in teaching that class, I had some of my own memories of what it was like to be a student in that class, and that helped me to shape a little bit more how we taught it. And I would say one example of that is including a lot more problem solving into the lecture itself to model some of the thinking that goes behind. How do you know which formula to use or which information is important or not important in a problem? By stopping and asking them to work on a problem before I worked through the solution, right? Giving them time to sort of think through it helped me, helped them, and seemed to be one example of one of the changes I made to the course. And I should say very carefully that those course changes that we made in genetics were in collaboration with Georgi Chankovsky, who inspired a lot of it.
You said when you were in Greg's lab that part of you identifying that the pattern of inheritance was because of the coursework. Not quite as great as being in the lab doing an experiment, but with that many kids, that's the next best thing. So it's kind of similar to trying to put together the applied versus the lecture materials.
Exactly. Yeah.
So what about trying to engage that many students? Because I liken it to a gladiator amphitheater where you're at the bottom and there's 400 people listening to you talk.
Yeah, so I've refined my approach to that over the years. I'm not somebody who I think is naturally outgoing, but I've come to terms with my role in the classroom. One of the moments that I dislike the most in that large, I think gladiators are great, description is those first few minutes of class, right? Students come in and their mind is on other things and they're talking with their friends about what they did last weekend, or they're shuffling through notes or something. And then you need to get 400 plus students to be quiet and focused and ready to think about genetics. And one of the tools that I've adopted, I guess, for doing that is short 1 to 2 minute video clips. And these might be something from a local news media or something more professionally produced by the Howard Hughes Medical Institute, or a silly YouTube video about the science of the day. And so by starting that, I think that students, hopefully it's engaging and interesting material. It's also a very clear sign that the class is about to start. And it gives me a way to connect the more basic principles to something that's more, I guess, real world applied. The more I think I can get from them, the better job I can do teaching them. And so I definitely view teaching in general as an experiment every time I try something new, right? And so I need to collect data and feedback and then refine it and try it again, the next year sometimes. The first time I taught genetics, I did something that my husband, again, who was a high school teacher at the time, described as masochistic. I decided that I wanted to know from students for virtually every topic in every lecture what was working and what wasn't. And so I wrote this survey that was completely anonymous about, what was the hardest thing to understand today, what was the most interesting, how was the time spent on these various topics that were, customized for each lecture. And the first time I taught, there were 300 students. Wow, I learned what I was not doing right quickly. It was not fun, I have to say, right, to get a lot of negativity twice a week when at the time I was putting 30 hours into developing one lecture.
As a junior professor, as you're trying to start your lab?
It was painful. The truth is that the reason I put a lot of time and energy into my teaching and tried to refine it and find things that work was because I wanted to do a good job. So I started with that, and I did what I thought was a good job.
Yeah.
I solicited the feedback that made it clear that some of that was not working. And after I got past the pain, I found ways to be more constructive and more effective as a teacher. And so a lot of my investment in time and energy was to avoid my own pain of going through that. Each semester, each lecture, I continued soliciting feedback through those surveys for the first 2 or maybe 3 years. And after a while, the response rate went down and the comments I got were pretty positive. And that's how I sort of knew, okay, maybe we're okay, right? Maybe I have a strategy that's working. Yeah.
Well, I have a million more questions, but I know we're out of time. So I really want to thank you for taking the time and having this really great conversation.
My pleasure. Thank you, Monica.
Thank you. Bye-bye.
Yeah, bye.
So if you're enjoying finding out about life beyond the beaker and discovering that scientists are not robots, but real people, help us spread the word about How to Science by leaving a review on Apple Podcast. Go on Twitter, #HowToScience. Tell people you like it. Tell us if you liked it. Tell us what you want to hear next. Thank you to LSA, the Liberal Arts College at the University of Michigan, for being the force behind the podcast and really doing all the work. Thank you to WCBN-FM, the University of Michigan's campus radio station, for letting us use their cozy, grungy space to record the podcast. Thank you to the Department of Molecular, Cellular, and Developmental Biology, where I spend most of my science time. Thank you to the Liz.
Find the How to Science podcast on Stitcher, Apple Podcasts, or lsa.umich.edu. That's lsa.umich.edu. Or wherever else sound is available on the internet. Visit weekly for new episodes in the series. Podcast theme music by Poddington Bear. Other music by Ant the Symbol. How to Science with Professor Monica Deuce, who says,
I can't stop eating cookies.